Hello Year 11, it is Mrs Shanahan here again, and this particular question, which is for paper 1 on Tuesday, is higher only. So if you're doing foundation, you don't need to watch this video. So this is quite a common question. It says bacteria have been genetically engineered, so this is from topic 3, to produce human insulin since 1978. Explain how bacteria can be genetically engineered to produce human insulin. So we've got to obviously talk about this process of genetic engineering involving bacteria to produce human insulin. So just a reminder, just some quick notes. When you're talking about bacterial cells, which are prokaryotic, and that means that they don't have a nucleus. Sometimes they can have multiple flagella, which are tails for movement. They have these circular plasmids, and then they have this sort of single chromosome here. So it's this plasmid here that is removed and used in the process of genetic engineering. When you are removing any gene, so the human insulin gene, that would be taken from a particular chromosome, and enzymes are used for the removal process. So to just go through a rough idea of what you'd need to be writing about here, first of all you would need to remove whatever gene it is, so even if it isn't insulin, whatever the gene they're talking about in the question, so remove the gene for making human insulin, or insulin if you're posh. I've always called it insulin, it doesn't really matter, and you would use restriction enzymes. So that is a key word. So remove the gene for making human insulin using restriction enzymes. And then you would need to remove the plasmid from the bacterial cell. So you use something called lysosomes or lysozyme for that. So you would then remove the plasmid that you're going to use from a bacterial cell, and that is using lysosomes. So you've basically got now a plasmid. We're going to have to open it up, so we cut it open. We need to make a bit of a gap there to put the human gene in, and again you would use restriction enzymes. So you would open the plasmid using the same restriction enzyme that you've just used to take the human gene out, using the same restriction enzymes. So basically you've now got a gap in your plasmid, and you've got your human gene that you've taken out. That's the worst diagram of DNA ever, but anyway, you've taken out your section of DNA, which will be your gene, and you want to be able to insert that. Now what you end up with are what are known as sticky ends, so unpaired bases of DNA. So just say, for argument's sake, that's an A adenine, and this side here it's T for thymine. These will have sticky ends. So that one there you would have T, and this one here you would have A, and they would join together. And you can call them sticky ends. So we've removed the gene that we want to have the bacterial cells replicate. We've removed the plasmid from the bacterial cell using what are called lysosomes. We've opened up the plasmid, we've made a gap using the same restriction enzyme, and we now add the human gene and connect using what's known as ligase, which again is another enzyme. So that's another key word to use. And then you need to put that back into the bacterial cell. So you would place the, so we call it recombinant now. So because it's got DNA from another organism, it's now called recombinant plasmid. So back into the bacterial cell, and then of course for bacterial cells to grow, they need oxygen, they need warmth, and they need moisture. So you'd make sure those conditions were correct, and then as the bacteria reproduce or multiply, they make human insulin. So they read the genetic information as their own, and they make human insulin, which obviously we can use for diabetics. So people who are type 1 diabetics, their pancreas doesn't work, and they are given human insulin, which has been made by bacteria. Hope that makes sense. I know it's a bit rushed, but hope it helps for Tuesday.